Okay, let's talk about water and bases. So, you all have this in your study guide. Have a look where it's in a lot more detail. But what I'm going to do in our presentation today is I'm going to summarize all of the points and really help to kind of re explain it. So, you're not just reading something, but you'll actually understand water and bases and properties and pH and all of that fun stuff. So, let's just jump right into it here. So, first, we need to talk about properties of water. Okay, so water is a polar molecule, meaning it has a slight negative charge on oxygen and positive charges on hydrogen. So, think positive charge hydrogen, negative charge oxygen. Okay, so they're opposites. This polarity allows water molecules to form hydrogen bonds, which gives water many of its special properties. You can't have water without hydrogen, H2O. That's where we talk about the hydrogen and the oxygen. But then you have cohesion and adhesion. So, cohesion is when water molecules stick together due to hydrogen bonding, creating high surface tension. So, water striders kind of walking on water. That's a high surface tension area. But then you have adhesion, where water sticks to other surfaces, enabling capillary action, such as water rising in plant roots or narrow tubes. So, cohesion is kind of like sticking together doing due to bonding, whereas adhesion is sticking to other surfaces that it's kind of like attracted to. It's sticking to those areas. Now, high specific heat, this is when water absorbs a lot of heat before its temperature changes, helping to regulate body temperature and stabilize basically water environments. So, water absorbs a lot of the heat before the temperature changes in that water. If it's super hot outside, your water isn't automatically like the water in your cup isn't automatically going to be the same temperature as the outside temperature. It's not going to be stone cold, ice cold anymore, but it's going to slowly raise its temperature, but it takes time, but it's never going to be the same temperature as outside that cup. But then we have high heat of vaporization. So water requires a lot of heat to evaporate, making sweating an efficient cooling mechanism for organisms. This is how we get rid of our heat, which is by sweating. Animals, as an example, they don't sweat. Their heat is released by their mouth. So when they're panting, that's how they release heat. But us, we sweat. Sweating is a good thing because we need to release that heat to control our body temperature. But then we have a universal solvent. So, water dissolves many substances, including salts, sugars, and ions, making it essential for chemical reactions, nutrient transport, and waste removal in the body. So, these are all very important. This is why water is so important. And then you have pH. So for some reason, I love talking about pH because once you guys do become dental professionals, pH gets talked about a lot because we're constantly battling the pH of our mouth, the pH of liquids, foods, and cavity causing things. What forms a cavity? This is where pH comes in. But what is pH? So pH measures the concentration of hydrogen ions in a solution, measuring hydrogen. So this is the pH scale at the bottom here from acidic, neutral to alkaline. So the lower the number is more acidic, and then you have the neutral numbers in the middle, and then the more basic solution are the higher numbers. So again, low pH means acidic, high pH means basic, which also means alkaline. So make sure you know that basic means alkaline, and neutral is going to be 7. You'll see here 6, 7, 8 because 6, 7, 8, like 6 is like neutral acidic, whereas 8 is neutral basic, but 7 is neutral. Water, for example, is 7. Now, pH is essential for enzyme function biochemical reactions, and cellular processes. So, for example, human blood maintains a pH of 7.35 to 7.45. So, there's a very slight range there. It's more neutral. Whereas stomach acid is very acidic, it can go from 1.5 to 3.5. So, 1.5 being more acidic than 3.5. Saliva and bodily fluids, we have pH around 7.4. This is important. So, our saliva can change depending on what we're eating and drinking, all of that. So, acid and base reactions, what's the point here? So, acids are going to donate H plus ions, and then bases are going to accept these H plus ions or release OH negative ions. So sodium hydroxide as an example. So hydroxide is NaOH. But then you have neutralization, which is an acid plus a base, water plus salt. So HCl plus NaOH, arrow NaCl plus H2O. So salt, water, all of that fun stuff. Now, when we're talking about what is a buffer in biological systems, so a buffer is a solution that just basically buffs things out. It maintains a stable pH either by neutralizing added acids or bases. So it kind of buffers things out to bring them back to neutral or as neutral as possible. So, this is critical because many enzymes and processes depend on a stable pH to function properly. So, how do buffers work? They contain a weak acid, so not a strong acid, but they contain a weak acid and it's base that goes along with it. So, or a weak base, and then it's acid that goes along with it. When an acid, H plus, is added, the buffers conjunctate base reactions with it, forming the weak acid and preventing a large pH decrease, because the lower means the more acidic. But then you have when a base is added, the weak acid is going to donate the H plus to neutralize the base. And then we have an example here for you. The carbonic acid with the bicarbonate buffer in blood stabilizes that pH around 7.4. Now, what are the importance here? So basically, the bicarbonate buffer system keeps blood at a certain range because extreme changes can be a bad thing. We don't want it to go up and down. We want it to be kept very, very similar, similar to the same. But then cells and enzymes. So enzymes have an optimal pH. For instance, pepsin works best in acidic conditions, while trypsin works better in slightly basic conditions. So it depends on the conditions. It depends on the body, vice versa. It depends on inside the mouth. If your mouth is on the acidic side, it's going to be easier to get a cavity than if your mouth was on the neutral side or even basic side. It's going to be a lot harder to bring it back to the acidity side, which is what causes the cavity. Because if you have a number of 8 and to cause a cavity it's 5.5, well, that's going to be a lot harder to get down than if your mouth happened to be at a 6 and it just needs to get to a 5.5 pH to cause a cavity. So we are trying to maintain the buffer system so there's not too many changes, not too many regulations and things happening. So, buffers help to regulate pH in the intercellular fluids, the cerebral spinal fluid, and urine to ensure proper functioning. So, buffers are so important. Now, common buffer systems are the carbonic acid bicarbonate buffer, which maintains blood pH, the phosphate buffer system, which works in the intracellular fluid, and kidneys to maintain pH in cells and tissues. Kidneys are so important. And then protein buffers are proteins like hemoglobin that can accept or donate proteins, helping to always be maintaining that pH in the blood and the cells. Now, what is cellular respiration? So, this is when we talk about glycolysis. the Krebs cycle, and the electron transport chain. So just remember, glycolysis is in the cytoplasm. The Krebs cycle is in the mitochondria, and then the ETC, the electron transport chain, is in the mitochondria. So that's why you have to know that certain anatomy, if you will, because you need to know what's happening in our cells. But what is cellular respiration? So, this is the process by which cells convert chemical energy from nutrients, mostly glucose, into ATP. As soon as you see ATP, think energy. The I can never say this right. The adenosine triphosphate. I should have just said that, and then you guys would have been none the wiser that that's probably not the way to pronounce it. I'm a dental hygienist, I'm not a scientist here. But then, which cells use for functions like muscle contraction, protein synthesis, and active transport? So, this can be with oxygen or without. It does not need oxygen to survive, it could be without oxygen. So, aerobic is with oxygen, and anaerobic is without oxygen. So, the overview is those three main stages the glycolysis, Krebs cycle, and electron transport chain. And then there's some chemical equations here for you to memorize these. So let's first talk about glycolysis. Again, located in the cytoplasm, what does this mean? It's the breakdown of glucose. So this is the first step in both reactions with oxygen or without oxygen. It does not require oxygen and breaks down one molecule of glucose into that six carbon and then into two molecules of pyruvate, which is a three carbon. But these are the key steps, though. So, this is how energy is happening. So, 2 ATP are used to activate glucose. This is energy. And then glucose is split into two three carbon molecules. And then the three carbon molecules are then oxidized, producing NADH, which is the electron carrier, a carrier here. And then four ATP molecules are produced by substrate level, phosphorylation. And this is direct ATP production. The end products are the two pyruvate, 2 ATP, and then 2 NADH. So the glycolysis summary is basically does not need oxygen. It occurs in the cytoplasm. It breaks down one glucose molecule into two pyruvate molecules and produces a net gain of 2 ATP and 2 NADH. Something to memorize there. And then the Krebs cycle, though, what is this? So glycolysis is the first. The Krebs cycle is the second in aerobic respiration. This occurs in the mitochondria, where pyruvate from glycolysis is even further broken down to produce more ATP, NADH, FADH2, and CO2 waste product. So this is called the citric acid cycle because the first molecule formed is citric acid. This could be a question on your exam is what is the Krebs cycle or what is the citric acid cycle? You know they both mean the same thing. Some call it the citric acid cycle because it's the first molecule formed. But the key steps here are the pyruvate conversion, the acetylcholine then enters the cycle, and then the citrate is oxidized. So all of these reactions are happening. In the first part, the pyruvate conversion, there's a two carbon molecule. And then you have the four carbon after that to the six carbon. And then you have where the citrate undergoes enzymatic reactions, losing waste products. So to CO2, high energy electrons are now transferred to the NADH and FADH2. And then the ATP is now produced by a substrate level phosphorylation. Okay. So the four carbon molecule is regenerated, ready to combine with another acetylcholine. Basically, okay? So What does all of this mean? It just means certain things are being converted and combined to produce an enzyme reaction. And then waste products are being taken out. All of this takes energy to create this reaction. That's all. Now, products of the Krebs cycle, to kind of summarize it even more, is the 6 NADH, 2 FADH2, 2 ATP, and 4 CO2. Something to memorize. I have it here in purple for you. Now, again, this occurs in the mitochondrial. You need oxygen, and then it's further broken down. So, this cycle is a cycle and it keeps on being regenerated to continue the whole process again. So, this is the Krebs cycle. Now, the electron transport chain, the last step. So, what is this? This is the final step in the aerobic respiration and occurs also in the mitochondria. It uses high energy electrons from the NADH and FADH2 to generate ATP, so more energy, electron transfer. So, the key steps here are that the NADH and the FADH2 donate electrons to protein complexes in the mitochondria. These electrons are then passed along the chain in a series of reactions. You have a protein gradient creation. This is when the electrons move through the chain using more energy and used to pump protons into the intermembrane space, creating a proton gradient across the mitochondrial membrane, allowing things to pass through. Pardon me, you guys. So then you have these protons then flow back through the ATP synapses there, and then the ATP uses this proton flow to produce ATP from ADP, an inorganic phosphate. So what's the role of oxygen here? At the end of the electron transport chain, the electrons combine with oxygen and protons to form water. This is how water happens. Oxygen is the final electron acceptor, making it essential. So, oxygen is needed. So, what are the products here? More ATP and water, basically. Okay. And then, so what's the summary? This happens in the mitochondria, requires oxygen, uses electrons from the NADH and FADH2 to create a proton gradient. The key word here is proton gradient in the electron transport chain. You don't have that anywhere else. And what is that again? Is it helps to convert that energy moving things around, and then oxygen combines with the electrons and protons to form water. A lot happening here, a lot. Now, again, you guys, a summary. So you have glycolysis, the Krebs cycle, and the electron transport chain. So think glycolysis occurs in the cytoplasm. The Krebs cycle and the electron transport chain occur in the mitochondrial matrix membrane. In glycolysis, again, the first step, it has to do with glucose, which is broken down into different molecules producing 2 ATP and 2 NADH. But then what is the Krebs cycle? This is where the pyruvate is converted into acetylcholine, which then enters the Krebs cycle producing 6 NADH, 2 FADH2, and 2 ATP, and 4 CO2. So glycolysis is the 2 ATP and 2 NADH. The Krebs cycle is 6 NADH, 2 FADH2, 2 ATP, and 4 CO2. The electron transport chain, then, what is that? This is where the NADH and the FADH2 from the Krebs cycle are going to donate electrons, creating that protein gradient, which I talked about, very important for the electron transport chain, that powers the ATP synthesis. Oxygen is the final electron acceptor forming water. So, to just kind of break it down a little bit more, again, glycolysis is 2 ATP, 2 NADH2. The Krebs cycle is 6 NADH2, 2 FADH2, 2 ATP, and 4 CO2, whereas the electron transport chain forms water, if you want to keep it that simple. Okay, so you might have been wondering, well, what does NADH stand for? What does FADH2 stand for? So these are, remember, electron carriers that are needed in especially the electron transport chain. Now, I'm going to try to pronounce them for you. So the NADH is the nictomide adenine diglucotide, and the FADH2 is the flavin adenine dinucleotide. These are hard to say. But you guys, that's what this is. So the NADH, the FADH, just think high energy electrons. Okay. And then the FADH2, it carries these electrons to the electron transport chain. It donates certain electrons because you're going to need that with the ATP. So, what's the point of all of this? So, both this NADH and FADH2 are essential. It's needed for the final stage of cellular respiration because they provide high energy electrons. We need high energy electrons to do the work that are used to generate ATP through oxidative phosphorylation. Okay, so think energy. All of these things are happening all the time, all the time. Now, something else to make note is what if there's no oxygen? So, what if no oxygen is available? Cells are now going to switch to anaerobic respiration or fermentation. So, in this case, glycolysis still occurs, but the pyruvate is converted into either lactic acid in animals or ethanol and CO2 in yeast with no ATP generated from the electron transport chain. So, again, when no oxygen is available, cells have to now switch to an anaerobic respiration. No ATP is going to be generated as there usually is in the electron transport chain. So, the anaerobic respiration produces far less ATP than the aerobic respiration. So, what did you guys think of that? Let me know if any questions. I tried to break it down for you in the dental hygiene admissions study guide that you have on waters and bases and cellular respiration. I break it down actually even more. There's more points for you, but this presentation was just mainly to kind of summarize it for you, help you study it, help you learn it, and then let me know if you have any other questions.